Thanks. And actually, this isn't from the density management study. This is actually going to be the results of two studies that we ran sort of back to back, but with the same intent. They were stimulated by questions that arose under the Northwest Forest Plan and the efficacy of buffers in providing for desired ecological conditions. One of the books that's out there on the display table is a book called Mammal Community Dynamics by Zabel and Anthony, and there's a chapter in there that focuses on riparian associations with mammals. And in that chapter, they cite something like 147 species of mammal that occur in Oregon and Washington, and of those, 95 have some level of association with riparian systems. Of those that are associated with riparian systems, they classified them into whether they're obligates, that is, their life history really depends on riparian ecosystems. Things like the water shrew would fall into that group. Then there are some where studies have shown a statistically significant association compared with non-riparian sites. And of those 95 that are riparian associates, 34 or so are in that associated category. And there are some that occur in early cirral forest associated with streams. That's a very small number, 5 or so. And then there are a bunch of species, over 50, that might be found in riparian systems, but which don't have any particular association with them and just seem to use them randomly as forest that may or may not be associated with streams. Well, over 10 years ago, we started one study where we just looked at existing conditions to see if we could investigate whether buffers were providing desired conditions for small mammals. So this was a retrospective approach to searching for existing examples of buffers, non-buffered, old and young forest, and comparing small mammal communities among those sites. We found 49 sites distributed through the Olympic Peninsula, and we looked at stand level responses of small mammals to various levels of harvest activity or no activity in streamside habitats in those streams. And these were all headwater situations. And then the idea was to compare abundance and diversity of these species in both buffered, unbuffered, and other management situations. Here's an example, 2 sites on private lands in the southeast Olympic area, showing the narrow buffers that were associated with clear-cut harvest everywhere else. The stand conditions that we evaluated were old forest, unmanaged old forest, mature forest, young forest, intermediate aged, then old forest that had these narrow kind of buffers or some kind of buffer on them, mature forest that had some kind of buffer, and then mature forest that had been thinned. We actually were studying not only mammals, but birds and amphibians and a number of other organisms, fishes and invertebrates, but I'm just going to focus on the mammals here. And for the mammals, we had 2 arrays of traps, one right next to the stream and then one at an adjacent upland site away from the stream. In the few years that this study went on, we had 768 captures, over 13,000 trap nights. And when you look at total abundance of all the species together among these different conditions, the x-axis shows the different stand types, and the numbers above those labels are the numbers of streams in each of those stand types. We found, just as everybody before me has reported in these studies, high variation in the results that we observed. And you can see there are some slight differences among these means, but the variance among the sites that we sample is so large that you really can't say there's any kind of a difference in total abundance among these stream types. Now, when you look at individual species, for instance, the montane shrew, which is one of the riparian associates that Anthony et al. Listed in their chapter, there is a suggestion of some sort of response here. The two, you look at buffered mature versus mature, you'll see a decline in abundance in the buffered situation. And the same with buffered old, where they didn't occur at all, versus old without buffers, where they were somewhat more abundant. There's a suggestion of a response, but again, these variances are so great that you can't say there's a real strong statistical difference. As we looked into this, it really revealed the shortcomings of a retrospective study in that there were all these confounding variables that we weren't controlling for. There were things like the stream gradient, the elevation, the substrate condition. And when you look at how many of these factors, or how many species responded to these various factors, including stand condition, stand condition, which is the first area here, which is this one in here, none of the mammals actually showed a strong response to stand condition, and that's the condition that included the buffer or not. What they did respond to were those things I mentioned: stream gradient, elevation, parent rock, and other aspects of the vegetation surrounding these sites. So one thing that was immediately obvious to us was when we looked for buffers, those buffers occurred where there had been harvest, and there's non-random distribution of harvests over that area, where most of the harvest and logging occurred at lower elevations along wider streams with shallower gradients. So there were some built-in biases that really influenced, we think, the results of the study. So the conclusions from that early work were that buffered and intact streams supported pretty comparable assemblages, not only of mammals, but of fishes, birds, and some of the other features. Amphibian density did vary quite a bit. Management, like I mentioned, was confounded with elevation and gradient. Variances were large, and rare specialized species weren't sampled adequately to draw meaningful conclusions. So that led to a thought that, well, if we could just get involved with an experiment where some of these confounding variables would be more tightly controlled, we would have much better success. And that led to the second phase, which was a riparian ecosystem management study, but using an experimental approach. And in this case, we were working or partnering with the Washington Department of Natural Resources, who were willing to put in these experimental treatments. So the overall design of this study, the idea was a before-after control impact, a Bakke design, where we had pre-treatment sampling, followed that with post-treatment sampling, and then we had treatments within adjacent catchments so that stand and topographic conditions were fairly closely matched. And those treatments were a control with no buffer at all, I mean no harvest, zero buffer where they cut right up through the stream, a continuous buffer, and this was under forest practices, and these were narrow 50-foot buffers generally, and then patch buffers where the buffer came and went depending on site-specific conditions. So if there was some unstable slope or a seep area that was desirable to buffer, buffer was placed there, but in other parts there would be no buffer. These treatments were replicated in 2 geographic areas, and that was in the Capitol Forest, where we had 4 blocks of these treatments, and then down in Pacific County in the Willapa Hills, where we had 3 sets of treatments. And just as an idealized schematic, this represents a sort of cartoon version where we have the fixed width buffer, as I mentioned, generally about 50 feet wide on each side, following the length of the stream. A variable width buffer that's we call patch buffers, where there may or may not be buffer along particular sections of that stream. And these streams, by the way, are just like the ones that Dee Dee had talked about earlier. Most of these were permanently intermittent. These are very small headwater situations. Then we had the no buffer treatment, where there was no trees retained whatsoever, and then a control where it was left alone. Here's an aerial photo illustrating one of these sets before and after treatment. So here's a pre-treatment scene in 2003, and then you can see the control stream up here with the forest left intact. And then we have a continuous buffer, which extended the length of the stream right up in here. And then an example of a patch buffer, where you see these coming and going sections of retained trees. And then the no buffer situation right down here. For small mammal sampling, we use live traps. Because these buffers are so small, though, there's only a limited size of trapping array that you could place within that buffer situation, which is one of the limitations of this sort of work with larger animals like mammals. But these traps were spaced fairly closely, kind of on a 2 by 5 meter zigzag interval on each side of the stream. There were 18 traps, 36 total, 18 on each side. And then we checked the traps every day for 4 days, 3 nights. We did this 2 to 3, or we did it for 5 successive bouts, 2 to 3 weeks apart. just as we found in the retrospective study, if we look at total abundance of small mammals among these treatments, and especially compared with the control sites, we really saw no difference overall. I'm not showing the variances in this particular slide, but the variances were still very large, even though we had controlled for many of these other variables. And none of the buffer types showed a markedly different total number of small mammal species, or of individuals. Now, we did see differences in community structure among these treatments. I need to walk you through this a little bit to show you what I'm talking about here. Over here in the control, this represents the percent similarity of species among sites within any of these treatments. In the control, this is the percent compared with the pre-treatment numbers. So in 1 year following treatment and then 2 years following treatment, there was roughly 55 to 60% overlap in species composition. So even in the controls, there was a great deal of turnover in species occurrence within the sites. Now in these various treatments, the strip transect, which is that continuous transect, the patch and the no buffer treatment, we found roughly comparable levels of similarity. In this case now, it's compared with the control, not compared with the treatment year. So in any one of those bars, what you see is the percent similarity with the equivalent year in the control data. The only statistically significant difference was in the absolute no buffer treatment, and there the similarity dropped drastically after the second year following logging to where it was only about 30% similarity between species composition in those no buffer treatments versus the controls. Now I'm just going to illustrate some of the reasons for that lack of similarity by going through some of the species examples. Peromyscus keenai is the northwest deer mouse, and it's a close relative of Peromyscus maniculatus, which is the deer mouse, one of the most abundant and generalized small mammals we have. So this is an animal that would have been classified as a non-riparian associate, but it's an obligate tree dweller. It's semi-arboreal, and it showed a very strong decline relative to controls as time progressed following treatment. And that probably reflects the loss of tree cover that they're associated with in those various treatments. So this one had a fairly strong response to the creation of those buffers. Now, this guy is the redback vole. It is often associated with older forests, but sometimes with young forest if it has enough downed woody material. They really have a strong association with logs and downed wood. There were high densities of logs and downed wood that were created with these buffer treatments. And for this species, we see a pretty strong increase in numbers of these animals over time, especially in the no buffer and the patch treatments compared with the controls. So this one increased in abundance. Another one that increased is the Townsend chipmunk, and it's another loose riparian associate, probably would have been classified as non-riparian because it's really a forest dweller, also semi-arboreal like the northwestern mouse. And it also increased in abundance following treatments. You notice the strong increase compared with pre-treatment. Oops, I'm going to learn this. The pre-treatment again is the black bar. The next two are the 1 year following treatment and 2 years following treatment. And you can see a big increase in post-treatment numbers. The Montane shrew is the one I showed from the retrospective study. This is one that does have a riparian association, a pretty strong one from other studies. And at first glance, you say, oh my gosh, that one really dropped following treatment. But if you notice, that drop also occurred in the control. So here we see a similar pattern in all the treatment types, including the controls. And so its changes in abundance probably reflect other factors that were going on that had really little to do with the treatment types themselves. So even though this one changed, it didn't change because of the creation of the buffers. So our key results from this effort, like I mentioned before, total abundance really didn't differ significantly among the treatments, but diversity changed, with some species increasing and some decreasing relative to numbers in the controls. Differences in this case seem mostly to do with changes in forest cover, not riparian conditions that were changed by the buffers themselves. But still, just like we found in the retrospective study, variation among these sites was really great, and that made differences difficult to detect. Some of the sources of variation, even though we had this kind of idealized buffer design, when they were implemented, the buffers really looked different, even if it was the same type of condition. So these are two illustrations of a fixed width buffer, but in one case, the forest was fairly dense along the length of the buffer. Interior conditions were maintained within the buffer. In the other, there were lots of openings, stand density was lower, and it was a highly variable and open situation, which resulted in different habitat conditions for the animals. The other thing is that after these buffers were created, we experienced a lot of wind throw, and some of these were exposed to strong winds that resulted in a breakdown of the buffer structure. So even after the first year of the study, we saw changes in buffer condition that resulted from wind and disturbance. So in the words of our friend, it is always something. If it's not one thing, it's another. In this case, the assignment of treatments we thought would be random and really idealized, but they weren't. There were some logistical considerations that the DNR went through. For instance, the control plots, they didn't want to run a road at great expense through an area that wasn't going to be harvested, so they saved the control plots for the places that would be hardest to build a road into. So the controls were not randomly assigned. And sometimes other of those treatments weren't randomly assigned for logistical reasons as well. The design wasn't as balanced as we had hoped. Not all treatments were replicated in all the blocks, so there was some variation in sample size there. The treatments didn't happen when we originally had hoped or planned. They originally called for a 2-year pre-treatment period and then 2 years at least of post-treatment, but it turned out that the market conditions were really good after the first year pre-treatment, and the buyers of the timber that DNR were using just had to get in there and harvest right then to take advantage of market conditions. So we only had 1 year of pre-treatment data. And then many of the species we would like to have studied are just too rare in this situation to sample with adequate frequency to make reliable inferences. So several of what would probably have been riparian obligates are also quite infrequent and would require completely different study methods in order to really understand their response to these treatments. So despite our hopes for the magic of an experimental approach, variation was really large and patterns were difficult to detect. So I'd like to thank the DNR, though, for working with us on this study, especially Richard Bigley and Jeff Rickliffs, who worked tirelessly to make sure that we could carry out the work. We got a lot of statistical advice from Jim Baldwin, who's a statistician with the Pacific Southwest Research Station, and the Pacific Northwest Research Station supported us with funding. And Pete Bisson and his co-workers were invaluable colleagues through the whole process. And then, of course, we thank our field crews. And then I'm citing one paper that you can look at for the details on this study. Thank you.
Any questions for Marty?
I'm just real curious about your variable width buffer treatment, where you have those basically areas with no buffer adjacent. Did you have enough variability in your data in terms of the spatial distribution of distances between patches to learn anything about how those organisms are using those patches?
No, we didn't. And I think that's the case where we'd want to use different methods. So just laying out an array of traps wouldn't tell us enough about those kinds of movement patterns. That's where really some more audicological studies of individual species with, say, radiotelemetry or more detailed mark recapture would have really been handy. So no, I can't say anything about movement among or between those sorts of patches, unfortunately.